So this method can help answer key questions about the ability of estrogenic ligands to regulate estrogen receptor alpha and the mechanisms of selective estrogen receptor modulators. The main advantage of this technique is that it is a simple method. It demonstrates ligand-dependent estrogen receptor alpha ligand-binding domain dimerization activity in the cells. Demonstrating the procedure will be Tanner Jefferson, a postdoc student from our group.
This method uses 3 different plasmids to detect the estrogen receptor ligand-binding domain dimerization activity in cells. PG5-Luc is the luciferase reporter plasmid for detecting the efficacy of dimerization activity. P-bind mouse estrogen receptor alpha-EF is a galactose-responsive transcription factor, Gal4 DNA binding domain fused mouse estrogen receptor alpha ligand binding domain expression plasmid that binds to the Gal4 responsive element on the PG5-Luc reporter. P-act mouse estrogen receptor alpha-EF is a herpes simplex virus transactivating segment protein VP16 activation domain fused mouse estrogen receptor alpha ligand binding domain expression plasmid that does not bind directly to the PG5-Luc reporter. When the proper ligand is present, the proteins derived from the P-bind mouse estrogen receptor alpha-EF and P-act mouse estrogen receptor alpha-EF plasmids bind through the estrogen receptor alpha ligand binding domain. The efficiency of the dimerization is determined by the luciferase activity. The most important step is controlling the activity of the P-bind mouse estrogen receptor alpha-EF derived protein.
The activity